Yo, welcome back to the Nucleogenesis Project. If you don't know who I am, my name is Dr. James Utley, and I'm here to be your resident codebreaker data scraper. Nucleogenesis Project is part of Syndicate Laboratories, as you know. This is part 5 of the video series. So, first off, let's just start off with what is nucleogenesis. So, the Nucleogenesis Project is a temporal framework for eukaryotic nuclear blueprint archival and reconstruction. So, what that means is developing a nucleus, a nuclear blueprint manifest of a nucleus that then can be in the future sent to synthetic biologists to then be able to reconstruct the nucleus. Okay, so this is, as I mentioned, this is part 5, so we're just going to get into it. Part 5 puts us right here at the fraction specific processing controls. So if you don't know where we are at, just go back and watch the other videos. That's an abstract, the primer, part 1, part 2, this is part 5. So here we go. Fraction specific processing controls. So as you know, we have two fractions. We have a test right now, biological fraction, and we have a test for the future, dried blood spot preserved fraction. All right. So we're going to start with fraction processing, right? So fresh biological fractions must be processed within a well-defined temporal and thermal constraints to preserve nuclear architecture and the labile molecular states. Unlike sequence encoded information, which is chemically stable, which is a chemically stable architecture. This architecture is dependent on features that degrade rapidly ex vivo due to the continued enzymatic activities, chromatin relaxation, RNA decay, and progressive loss of nuclear integrity, right? So this is the stuff you got to test right now, and this is what we know of to this date. Right. So these processing windows below are defined right here. So we have our high C, our ATAC seq, our cut and tag, our single cell RNA sync, our whole genome sequencing fresh. And then we have bisulfite sequencing here. And we have our maximum delay. So right now we have to be like right off the get, our ATAC sync and our high C, we got to get those all tested right away. The cut tag got to get tested away. This is the specimen type. So EDTA tube, so purple top, and then the fresh heparin is green top. And this is the temperature in which they should be stored at, within this window, which is basically refrigerated. All right, moving on. Okay, so these processing windows reflect the differential sensitivity of each assay to for post collection degradation. So chromatin conformation, or Hi-C, the testing mechanism, and chromatin accessibility, that's the ATAC-seq, are particularly time sensitive, as even short delays allow nucleosome repositioning and relaxation of higher order folding. So you're just trying to do your best to capture the information as soon as possible from a fresh sample. Single cell RNA sequencing exhibits the narrowest tolerance, as transcriptional profiles are rapidly altered by stress response and RNA degradation once cells leave the physiological conditions. So once the environment becomes unstable, the transcriptional profile is very difficult to catch. So you try to test that one as swift as possible. Right? So all fresh fractions are maintained at a refrigerated state to suppress enzymatic activity and slow the molecular diffusion without introducing any cold shock artifacts or compromising the nuclear integrity, right? So you're trying to keep it at like a a slowing state without being too abrupt. All right. While some assays, such as whole genome sequencing and bisulfite sequencing, tolerate longer delays due to their resilience on a chemically stable DNA, because the other is more it's an architecture and it has RNA and transcriptional profiles, right? So very unstable or very fragile. They are included. So the chemically stable DNA are included to emphasize that even sequence focused assays benefit from controlled handling when fresh material is used. So basically, it's like keep the standard on the fresh material, so you can test it as quick as possible. But you're going to have to test. There's going to be kind of a order of march, if you will, for the testing, because you're going to have to try to capture as much information in the smallest amount of time. So failure to adhere to these windows does not merely reduce the data quality, but results in irreversible loss of high entropy structure dependent information. So if like you don't follow this, it's not like you're going to get a worse set of data. You're going to get a data that is the data is irrelevant. It's non-existent. It doesn't matter. It won't do anything. And so consequently, that's why you have to adhere to strict temporal control of fresh fraction samples. And it is a prerequisite for any framework that seeks to integrate architectural priors with long-term archival genomic data. So for you to be able to really realize nucleogenesis as a framework, the fresh samples, you have to follow the rules exactly, because that is the basis. And we're, as I mentioned in other videos, we're building an anchor. So we have to follow as much of a static process as we can, right? Okay, so we know that we test the stuff right away in a very controlled format to build us a information layer of fresh samples. Now we have this archival protocol, which means like taking samples that can be tested in the future, and DBS cards. They also need to follow a standardized deposition and storage protocol designed to preserve chemically stable nuclear information, that's sequence and methylation, while generating a verifiable tamper evident provenance record. So we got to have the record to say this is what it is, and this is who it belongs to, and all the cryptographic stuff that we had mentioned in prior video. And then you have to follow the steps, because you're basing future actions off of the understanding of prior actions. Okay, so the protocol treats a DBS card not merely as a collection medium, but also as a long horizon biological ledger whose integrity depends on controlled drying kinetics, which is very important, moisture exclusion, no moisture, stable storage conditions, and continuous environmental accountability. So you have to understand all of these, and I have it written out here in this next table. But we have to know that the post-deposition degradation is dominated by hydrolysis and oxidative chemistry. Those are the single most important controllable variables in a residual moisture according to this protocol. It is, moisture first by design, because we know that will change everything. So with that said, we'll just circle back one time here. We know we have to test the fresh stuff as soon as possible, following the protocols that have been outlined in terms of the testing and the storage, meaning temperature of storage and the testing time and the tubes. Okay. Secondly, is now we have the archival protocol, which is the DBS, like so impregnating the DBS card, and we need to make sure that we do this in kind of a standard uniform mechanism, so we can then rely on it for future activities. So we have the event context, which is, and then we have the ambient drying phase, follow that, verify the desiccation for the moisture threshold to support hydraulic damage and the micro growth. We're going to follow that, the proven sealing, the barrier packaging, the storage and monitoring. So we know that if we have an environment for both frozen and controlled ambient storage, we should use it, but we should also make sure that it is robust enough to be able to survive if the freezing portion is not, if you can't do it because of whatever reason, right? Meaning like that there isn't the infrastructure that would support a robust freezing network. Okay. So the rationale and interpretability. This protocol is engineered around around an asymmetrical environment. Once residual moisture persists in the matrix, time-dependent chemical damage will accumulate. And in contrast, once the card is sufficiently desiccated and sealed, sequence and many covalent epigenetic marks remain recoverable over long horizons. All right, well, let me say that again because I was kind of mincing it up. The protocol is engineered around an asymmetry. Once residual moisture persists in the matrix, time-dependent chemical damage accumulates. In contrast, once the card is sufficiently desiccated and sealed, sequence and many covalent epigenetic marks remain recoverable over long horizons. Okay, so hopefully that makes sense to everyone. We're gonna turn the page here, and we're gonna get down to the chain of custody cryptographic protocol for next time. But before we close out here, the cryptographic fingerprint does not protect the biology directly. It protects the identity and the continuity of the archive by making later substitutions reliable or silent environmental excursions detectable in aggregate. The DBS archive becomes both a biological substrate and a verifiable integrity object suitable for delayed reconstruction workflows. So basically it says that the nucleosampler, or the mechanism to protect the identity and the storage of the data, doesn't necessarily protect the biology. And that is just kind of obvious. If you get it, if you understand, then it's obvious. Okay, and with that, We'll pause for today and we'll pick up our next section here, which would be part six. Part six is the next one, and we'll talk about chain of custody and cryptographic protocol. But here's a quick preview. It's a tamper evident audit trail. It's blockchain, block after block after block. And my name is Dr. James Utley, and I'll see you in the future. This is Nucleogenesis Project, and it's found at syndicate-labs.io. Syndicate-labs.io. All right. Peace.